All right, can you guys see my screen now, right? All right, thank you again, Jainu, for the kind introduction. And hello everyone. It's my great pleasure to be here today. Sorry, I changed the title. I want to talk about. We'll talk a gut feeling about barrier protection. And hopefully, after my talk, I can convince you that it is not just my gut feeling. I still have some data to prove that. I believe everyone here today at some point have the experience of abdominal pain, which is the feeling of discomfort anywhere in your belly, which is also called stomachache. There are numerous reasons for abdominal pain, but the main causes are like infection, abdominal growth, inflammation, obstruction, and also diseases like affect other organs in your belly. As you can see from here, most of the causes are related to the gastrointestinal system. Canonically, the abdominal pain serving as an alarm, meaning that when you have there's something wrong in your belly. But I'm very curious, except for serving as an alarm, does abdominal pain also contribute to the regulation of other functions of gastrointestinal barrier functions? So the first question is why we have gut feelings. Using the technique of ideal staining, you can see from here that the gut is densely innervated by neurons, and the neurons innervate all gut layers, from the muscle layer, mucous layer, all the way up to the epithelial layer. Now we know that the gut innervating neurons include automatic neurons like sympathetic neurons, parasympathetic neurons, and also enteric neurons located in the gut. And the GI tract is also innervated by sensory neurons running from the dorsal root ganglia or the nodose ganglia. Sensory neurons that are responsible for sensing various external danger signals such as heat, mechanical hurt, chemical hurt, or pathogen infections to mediate pain are called nociceptors. Nociceptor neurons can be marked by 9.8 and TPV1. Our lab and also other labs have now have demonstrated that nociceptor neurons innervate almost all peripheral organs, including skin, small intestine, and colon. But as to the role of nociceptor neurons in regulating the function of these peripheral organs is still largely unknown. The nociceptors, as I just said, it can sense external danger signals such as heat to be activated. On one end, the neuron activation triggered action potential can be transmitted via spinal cord to the brain, which makes us perceive pain. And on the other end, neuron activation can result in neuropeptide release, such as CGRP, substance P, and VIP, via which the neurons can modulate the function of the target cells. The crosstalk between nociceptor neurons with various immune cells has been widely reported in recent years. For example, the pathogen group A streptococcus and the fungi Candida albicans can trigger neuron activation and induce CGRP secretion, which targets on the downstream neutrophils and dendritic cells to modulate the whole defense. And the neuron secreted substance P can target to dendritic cells and mast cells to modulate type 2 immune response. Moreover, the neuron secreted VIP has also recently been targeted to RC2 cells to modulate the type 2 response. However, while the crosstalk with immune cells has been well characterized. Whether nociceptor neurons interact with epithelial cells is still largely unknown. Recently, there's a paper from our lab showed that gut-innervating nociceptor neurons can regulate M cells in the Pierce patch of a small intestine in the context of a salmonella infection to mediate the host defense against the pathogen infection. However, whether and how nociceptor neurons crosstalk with intestinal epithelial cells, especially at baseline, is still unknown. I'm very interested in the gut barrier, which is made of epithelial cells themselves, the perissolial junctions, and the mucus layer above them. Therefore, we can say the mucus layer serves as the first defense barrier that protects the host from danger invasions like bacteria in the lumen. Mucus covers the whole intestinal epithelial surfaces from the small intestine to the large intestine. Mucus are secreted from goblet cells, which is mainly transcribed from MUC2, the major mucus express genes. Therefore, mucus or goblet cells can be detected by MUC2 staining. Mucus or goblet cells can also be detected by UV1 staining, which detect those glycosylated mucus. In addition, Alcin blue and PS staining is also commonly used to detect mucus or the goblet cells in the gut, as you can see from here. In response to various triggers such as pampon molecules derived from bacteria like LPS, goblet cells can be activated to release mucus, as you can see from the red mucus from here. And here's a EM image of mucus secreting mucus. However, whether sensory neurons can also talk to goblet cells is still unknown. To address this question, we generated a nociceptor-specific nocturnal mice by crossing 1.8 Cre, which is a specific marker for the nociceptor neurons, with DT fluxby mice. With these mice, the nociceptor neurons will be specifically depleted. With these mice, we performed MUC2 and 16S co-staining, which labels the mucus molecules and the fecal bacteria respectively in the column. And here, the middle space between the fecal palate and the colon wall will be the mucus layer. From here, you can clearly see that the mucus layer in the nocturnal mice is significantly decreased compared with that of a control mice. Here's the quantification. To further confirm this result, I also used an ex vivo colon explant culture system, where the colon isolated from mouse will be opened and mounted into a double layer chamber system. As you can see from here, the blue is the colon explant, which is overlaid with the red fluorescently labeled 1 micron or 1 micrometer diameter, almost bacteria-sized beads. Due to the presence of mucus, the beads will be separated from the explant, and here the middle space will be the mucus, and you can clearly see that the mucus layer is dramatically decreased compared with dental control with much higher permeability, which this data suggests that the nociceptor abolition can lead to decreased mucus thickness. So the next question we ask, how about we activate the neurons? To this aim, we generated a chemogenetic nociceptor neuron activation mice by crossing 1.8 Cre with HM3DQ, the activation dread flux mice, where the neurons can be specifically activated by the drug single treatment. We confirmed this system using in vitro cultured DRG neurons, where we found that the CNO can trigger neuron activation isolated from the Nav1.8 dread mice, but not neurons from isolated from the control mice. With these mice, we also performed the ex vivo colon explant culture experiments and found that the mucus layer in the CNO treatment mice or the neuron activity device can be dramatically increased 10 minutes after CNO treatment. Since GOB cells are the mucus secreting cells, so we predict that the nociceptor neurons might have a close proximity to GOB cells. So indeed, here, as you can see from here, using the nociceptor reporter mice with combined with macro staining, we found that the nociceptor neurons are just exposed with GOB cells in the colon. So the next question is how nociceptor neurons regulate the mucus thickness. To this aim, we purified total epithelial cells from colon and performed single-cell secretion. As similar to previously published in the literature, the total epithelial cells can be divided into several different subsets, such as enterocytes, enteroendocrine cells, like stem cells, and also GOB cells. Since, as I mentioned, the nociceptor neurons can secrete neuropeptide to modulate the target cells. So we analyzed the expression of list of neuropeptide receptors that can be potentially expressed in this epithelial subset. Interestingly, we found that RAMP1, the receptor for the neuropeptide CGRP, is highly expressed in GOB cells, but not other receptors. And only GOB cells, but not other cell types that specifically express RAMP1, which is also shown by UMAP plot here, that the CGRP receptor RAMP1 is highly expressed in GOB cells. We also confirmed the expression of RAMP1 in MUC2 positive GOB cells using iron scope staining, which also shows that it is highly expressed in GOB cells from both mouse colon and human colon. So this data suggests that nociceptor neurons might regulate the mucus thickness via CGRP-RAMP1 signaling. Consistently, we found that with the mucus using MUC2 and 16S co-staining, we found that the mucus layer from both CGRP knocked mice and the epithelial cell specific RAMP1 knocked mice, both of them showed decreased mucus layer compared with their legitimate control, which phenocopied what we found in the nociceptor knocked mice. Consistently, we also found that while there's a higher expression of CGRP in the mesenteric nerve, where nociceptor go to the colon, there's almost no CGRP in the knocked mice. We also confirmed this using CGRP using ELISA, which also showed that both serum and colon displayed decreased CGRP expression compared with that of control mice. So then the next question we ask is, what are the stimuli that regulate CGRP production? Our previous study showed that the pathogen Salmonella can trigger neural activation to secrete CGRP. So we assume that maybe the commensal cues in the lumen can also contribute to neural activation and the following CGRP release. Indeed, we found that germ-free mice which lack luminal microbiome showed dramatically decreased CGRP compared with SPF mice. And the following experiments show that transplantation of SPF fecal pellet to germ-free mice can significantly upregulate CGRP. And the CGRP upregulation induced by fecal transplantation into germ-free mice can be blocked by RTX pretreatment, which is toxin that you commonly use to deplete nociceptor neurons, which means that the commensal cues can induce CGRP aggregation in a nociceptor-dependent manner. Consistent with this, with the CGRP regulation, we also found that fecal supernatant, which contains the commensal cues isolated from SPF mice, can trigger much more neuron activation and CGRP release from the in vitro culture DRG neurons compared with that triggered by the germ-free fecal supernatant. Inconsistent with the CGRP upregulation, we also found that the mucus layer is also increased after fecal transplantation from SPF mice to germ-free mice, and the mucus growth induced by fecal transplantation can also be blocked by RTX treatment, which demonstrates that the commensal cues can promote mucus growth in a nociceptor-dependent manner. Interestingly, we also found that capsaicin, the component extracted from chili pepper, can also induce mucus growth. So then the next question is how nociceptor neurons regulate and the downstream CGRP-RAMP1 signaling regulate goblet cells to induce the mucus growth. Since we observed that neuron activation can induce mucus growth as fast as within 10 minutes, so we assume that nociceptor neurons regulate the regulate goblet cells by promoting the mucus secretion instead of regulating the mucus synthesis or other process, since it only happens very fast, like 10 minutes. To test this hypothesis, we also generated a CGRP mediated activation rat mice and performed Alcin blue PS staining, which, as I mentioned, labels all those mucus-containing goblet cells, as you can see from here. With these mice, we found that 10 minutes after single treatment, almost all goblet cells in the colon are almost emptied compared with that of control mice. And the mucus and the goblet cell emptying induced by neuron activation can be blocked by BRBM pretreatment, which is a RAMP1 antagonist, which suggests that the neuron activation induced goblet cell emptying depends on RAMP1 signaling. We also confirmed this result in vitro using organoid-derived goblet cell cultures, combined with the aggravating orange staining, which labels all those mucus-containing granules of goblet cells, and MAC2 staining, which directly labels mucin molecules in goblet cells, and did a CGRP treatment, as you can see from here. Using both staining show that CGRP can trigger the goblet cell emptying, also in vitro. Accompanying the goblet cell emptying, we found that CGRP treatment can induce rapid mucus growth, both ex vivo using the explant counter system and in vivo using the MAC2 S16S co-staining. So then the next question is, since the mucus, as mentioned, is critical for maintaining the gut homeostasis, including the microbiota, So we performed the 16S sequencing, isolated from both mucosal and the lumen. Interestingly, we find that while there's no obvious difference to the mucosal microbiome, the lumen microbiome is dramatically dysregulated. Further analysis show that two bacterial families out of Firmicutes, which are Tyrosibacteria and erase blue trichia, as a blue color you can see from here, are significantly expanded. And out of these two families, we found two bacteria, Tyrosibacter and Allobacterium, are dramatically expanded in the knocked mice compared with that of control. We also wondered what's the effect of nociceptor ablation on goblet cells. So we also performed single cell sequencing of total epithelial cells isolated from the knocked mice, and compared with control, you can see there's no obvious difference to the cellular composition. And we also analyzed the expression of mucus-related genes. As you can see from here, most of the mucus-related genes, especially MAC2, there's no obvious difference, which again suggests that the nociceptor neuron regulates the mucus layer by regulating the mucus secretion instead of regulating the mucus synthesis. But we do observe more than 700 differential express genes of goblet cells between the knocked mice and the control. A geopathway analysis shows that most of these differential genes contribute to increased cellular response to virus, protein folding, incorrect protein folding, and ER stress, which means that the nociceptor ablation leads to increased goblet cell cellular stress. So then the next question is, what's the physiological function of the nociceptor goblet cell axis? Let's go back to the question I asked at the beginning. Abdominal pain or visceral pain is a typical symptom of IBD or the inflammatory bowel disease. And the DSS-induced colitis is a commonly used mouse model to mimic IBD. So we treated our mice with DSS, and by inserting a distinction balloon attached with a pressure sensor to the colon of the mice, we can detect their response to the balloon distinction, which indicate the degree of visceral pain. With this system, we found that while the control mice displayed remarkable visceral pain, the pain was almost completely abolished in the knocked mice. But is the pain abolishment we observed in the knocked mice good for their health? The answer is no. The following analysis showed that the knocked mice displayed much more body weight loss and have displayed shorter colon length, much more severe colon structure disruption, and much more goblet cell loss using the Elsing blue PS staining, which demonstrates that the neuron difference renders the mice more susceptible to DSS-induced colitis. Next, we also ask how about we activate the neurons. So we co-treated. To this aim, we co-treated the NAV1.8 direct mice with the drug to activate the neurons, and with the DSS simultaneously in the drinking water. And with this mice, we found that the neuron activation can protect the mice from DSS-induced colitis, as displayed by a better body weight, longer colon length, and also much better colon structure. So then the last question is, since it is the CGRP deficiency that that leads to decreased mucus thickness and renders the mice susceptible to colitis. What if we put CGRP back to the Nociceptor mice? To this aim, we utilized an osmotic pump system which carries CGRP and can continuously secrete CGRP after subcutaneous implantation. And we also treated these mice with DSS, and we found that the CGRP administration can rescue the Nociceptor mice from colitis, compared with the vehicle treated knocked mice. That is also displayed by both body weight and colon structure. So together, this data brought to the picture here that the Nav1.8 nociceptor neurons can sense various luminal signals such as commensal cues and dietary cues like capsaicin to be activated to secrete CGRP, which act on the X receptor RAMP1 that is specifically and highly expressed in goblet cells to promote mucus secretion, to build the mucus layer, which protects the host against colitis and also maintains the microbiota homeostasis. This work was published at the end of last year and also likely gets selected as a cover story. And here the cover image also showed that the flower here or the goblet cells here is nicely innervated by nociceptor neurons. At last, I want to thank my mentor as SF2. I'd also want to thank my close collaborator Amanda in the lab, who is also the co-author of this study. I also want to thank all the other 2 lab members, especially Kim, Liban, Samantha, Yulian, Glendin, Dylan, and all the other 2 lab members. I also want to thank my great collaborators, and also thanks for the grant support. And last, I want to thank you all, and happy to take any questions.
Thank you very much, Daping. That was a fantastic talk.
Thank you.
So yeah, if anybody has questions, please unmute and ask, or you can even ask in the chat. But while we're waiting for the first questions here, I noticed one thing that you presented, that capsaicin actually increased the mucus layer. So I would imagine it's like protective, but I wouldn't eat hot things when your stomach is aching. So it feels a little counterintuitive, you know? Do you have any thoughts about that?
It's a very yes, it's a very interesting question. So we do a thought about this question. So with this mice, we performed the capsaicin gavage. It do induce the mucus growth, right? Mm-hmm. And actually, based on the clinical study, then showed that people who regularly eat chili pepper, they do have a lower rate of mortality and mortality. So which mean that for those healthy people, it might be good for their health. But on the other hand, if you do have some intestinal disorders, it might be, worsen your disorder, yeah. It might do harm to because it also it triggers neuron activation. Once right, it might induce pain. I guess then something you don't like.
Yeah.
So I would say it depends on the context. I would say.
See.
Yeah, and also. Right. Yeah, and also, very interesting question. We are thinking to test what if we put like capsaicin in the mouse trial to see if it can protect the host. That's something we are considering. Yeah, thanks.
Okay, two more questions. So you talked about various bacterial increase or decrease in your receptor top downward all mice.
Sorry, I missed your talking because.
The microbiota that was increased or decreased. Is that same, right? The lumen microbiome.
Your internet is not connected, but I missed the main part of your question. Because you asked about the microbiota and what?
Yeah. So I'm asking, is there a significance in any particular strains that you see higher or low in the receptor knockdown control, or is it just an observation that you made?
I don't know, maybe it's my internet. I didn't get. So you're asking if the microbiota is changed in the let me explain the microbiota. Sorry, I still didn't get your quick question here, maybe because my end, I sit in the room, maybe the internet. So I just here I just show the microbiome analysis from the neuron optimized and the wild type at the baseline. We do observe the expanded several expanded maybe potentially opportunistic commensal bacteria. So and we are actually I didn't hear your question, so we also analyze the bacteria from the neuron activated mice, which also shows that the neuron activation can also help to stabilize the microbiome. Yeah.
I think that partially answers the asking is a significance in which strain is higher, strain is thought like that. Oh,
I mean, you're asking which strain is upregulated, right? Okay, we observed two bacteria, right? It shows the Tyrosibacter and the Helicobacter out of Firmicutes are significantly expanded compared with that control. And very interestingly, these two bacteria is also has been shown to be upregulated in the IBD patients. So, but we still don't know if they also contribute to like a colitis also, no.
So, Daping, I have another question. Thank you. So regarding the if you look at your singles or your RNA-seq data, you have some more like increased expression of type 1 interferon gamma genes. So I was wondering if these animals, if you challenge these animals with like an infection with, I don't know, I'm thinking of norovirus, which will infect the gastrointestinal tract, would you see a protection there? And that would be like nociceptor signaling negative for the protection, in particular in this context, because we only show in the microbiota context. I was thinking another type of context, like a viral infection.
Yeah, wonderful question. We know the norovirus is a very common like luminal infecting virus. And also type 1 is Visceral is quite important for the antiviral host response. But I mean, I need to emphasize the single cell sequencing was performed at baseline, and actually most of the genes contribute to increased cellular response. I assume this will make the mice, or specifically the goblet cells and also other epithelial cells, more easier to die, more susceptible to die, and various triggers like colitis we showed here. And also we tried some pest infection. It also induced much more cell death. So I didn't do that to the viral infection, but I would assume that it will also induce much more epithelial cell death, I would assume. because of the stress, and the increased top line response. I think is maybe due to the damaged mucus barrier that renders mice display the higher response. Maybe it will be interesting if we can detect if the virus, the genome of the virus is also dysregulated. That will be interesting. Yeah, but it's one wonderful question.
And I just have another question, if I can have the mic again. So regarding, I will take first Jainu's questions, and particularly on the DSS colitis experiment. So what would happen if we give a wild type animal capsaicin? We will see a protection there. And my question is, if the protection is only given by the mucus thickness, or is another signaling that will be happening not directly because of the mucus thickness, or maybe signaling within another immune population there, like maybe some indirect mechanism there. I don't know, like, for example, if you give, in the same context, you can give capsaicin to these animals that have depletion of these neurons to see the activation in other population within the gut, to prove that this is something directly because of the mucus thickness or other things happening there.
Mm-hmm.
Well.
Yeah. Good question. Here, actually, we indeed thought about that idea, to give the mice capsaicin. But the problem is sometimes we give the capsaicin, the mice, because it's chilly, the mice stop eating. They don't like the food. Okay. So they stop eat less food. So it makes the situation more complex. We don't know if the phenotype is due to the different eating, right, or the direct effect on neural activation. And on the other hand, I also do believe that except for the mechanism we showed here, the neurons can also talk to various other like epithelial cells or immune cells, like I did mention, like it can not just except for CGRP, it can also induce like a substance P or VRP. And if you notice from the single section, we do, although the expression level is not high, we do notice some level of VRP receptor expression in some other like enterocyte. But as to the function, we don't know. But we definitely can't rule out the other possibilities.
Okay. Thank you. Congratulations. Thanks. So yeah, if there's no more questions, I will thank Jainu and Daping for helping us today. Thanks, Vivian, and thanks everyone in the audience for joining today's seminar. Please follow our social networks, and this session is going to be recorded, and we're going to be uploaded to YouTube pretty soon, so you can watch it over again. Thank you so much, everyone.
Right. Thank you. Thank you, guys.